All right, so thanks very much for the invitation. So Ed invited me, so thanks, Ed. So today what I want to tell you about is some of the work we've been doing in the past few years on understanding how forces from the cell's microenvironment work their way through adhesion cytoskeleton to the nucleus and how that changes 3D chromatin organization. So to understand this, I just want to start with this slide where we look at different organs in the body. So the stiffest, of course, what gives the body its strength is the bone. The analog is a brick. The most important organ, arguably, is the brain. That's the softest, more like soft tofu that you scramble, right? And the stiffness is, that you span is about 6 orders of magnitude. Now you have cells in these organs, and the question is, how do these cells sense what's around them, and how do they adjust their signaling, etc. So where does this stiffness arise? That's a very good question. And so in collaboration with my colleague Paul Jamni, we looked at this. And long story short, in a tissue, you have both cells and the extracellular matrix. The stiffness actually comes from the extracellular matrix. It all comes from collagen, okay? Now, why is this important? Why is it important for chromatin organisms? It's actually very important, because if you take a triple negative breast cancer cell, then it has to start from where it is, where it's relatively soft. It has to make its way through the microenvironment, through the blood vessel, and then eventually end up in the bone, in which case it might be 2 gigapascal, really, really stiff. The same cell that is now seeing these 6 orders of magnitude of stiffness. And how do they rearrange their chromatin, rearrange their cytoskeleton, etc.? That's really the question. So our lab is part of the metastasis research network. So we've been primarily working on signaling, but signaling has actually led us into the nucleus now. All right, to understand how chromatin organization changes as the same cancer cell moves through the metastatic cascade. And the question that we're asking is, will a breast cancer end up in the liver, or will it end up in the brain or the bone? That's really the question. Very, very different mechanical environments, all right? So to start with, I have to tell you a little bit about signaling, because it's eventually the signaling that is leading to changes in organization. So what I'll tell you is, how do cells really sense their environment? How do they change their cytoskeleton? And actually, the organization of cytoskeleton has quite a bit to do with the shuttling of epigenetic regulators like HDACs, etc. And that's going to change the organization of chromatin in the nucleus. And then I'll go to the primary data that I'll be presenting here, where we'll be looking at cells sitting in a relatively stiff versus soft environment, and really through STOME imaging, look at how chromatin is organized. So really euchromatin versus heterochromatin. And the bottom line here is we see that you have these chromatin packing domains at about 100, 200 nanometers in length. And depending on whether you're sitting in a stiff or soft environment, there is a difference. The sizes of these domains are different. The amount that is stacked up against the nuclear lamina, LADs, that's different too. And we've also developed a theoretical model to understand that. And this is sort of put a cell and look at what's going on. We can also do dynamics. In the sense, you can actually stiffen these underlying substrates and see how the organization of chromatin changes in real time. So this is evolution in over 6 hours. So I'll tell you what's involved in this dynamics. So all of this is, so I come from a more continuum mechanics background. So all of this is sort of continuum modeling. But we've actually taken some of these insights and I've put it into polymer models, some of which developed by folks in this room. And actually we can see these packing domains. And I'll come to that very briefly. And there's a poster right there by our graduate student Vivek. So you can take a look if I don't get to it. All right, great. So how do you change everything that the cells, how do you keep everything that the cells sees constant, but only the stiffness? So where you do that is you take a glass slide, put a little hydrogel. Hydrogel is a soft material. And you change the thickness of the hydrogel with the same ligand preparation on the top. So what you will see is that the cell is saying everything is same except the stiffness, okay? And you can already see that the cytoskeletal organization is different. On a stiff substrate cells, these are mesenchymal stem cells. All the cells in the mesenchyme evolved from these stem cells. You actually see that the cells are more elongated, and more importantly, there are adhesions at these end and these cytoskeletal fibers that actually pull, and they actually squeeze the nucleus down. So here the nucleus is more like a pancake. On a soft substrate, the nucleus will be rounder. I'll show you more images in a second. And now what we've done in collaboration with Malike Lakarmiali and Rob Mock, so I direct the Center for Engineering Mechanobiology at Penn. So we hired Malike primarily to do all this imaging, high resolution imaging. She never worked on mechanobiology, but now we can't think of a center without Malike. So here what we do is, here we put the mesenchymal stem cell on glass. Here it is stiff, here it is soft. And this is stome imaging. And what you're seeing here, and here we are imaging H2B. So these red regions are where the, where chromatin is condensed, and blue where it is not condensed. And more importantly, what you see is that in the soft substrates, you have these large interior domains, and quite a bit of the chromatin is stacked up against the nuclear lamina. Now, if you're coming from a theoretical point of view of mathematics or physics or engineering, the question is, some characteristic size scales here. Where do they come from? And also labs have some characteristic size scales. Where do those come from? And when you reorganize, how does this reorganization take place? So that's basically what I'll be telling you about. So before that, very quickly in 2 slides, I will tell you about signaling. What does the substrate do? Okay. So this is the cell membrane. That's the outside. Oh, that's the outside. That's a trap. All right. So that's the that's the outside of that's the inside of the cell. So in response to the forces that the cells see, what the signaling machinery will do is it'll upregulate phosphorylation of myosin. So on stiff substrates, if the cell sees something stiff, it will pull harder. That's what the cell will do. Okay. So long story short, we've done looked at a lot of problems that have to do with cancer cell migration, immune cell migration, etc. But here, what's important is if the cell is on a stiff substrate, it's typically elongated. And you start off with isotropic contractility, but now because the shape is anisotropic, you'll generate an anisotropic stress. As a consequence, you'll start increasing contractility in that direction because of signaling. So here, there will be a lot more stress fibers, a lot more contractility here, much less contractility. And we can sort of compare with confocal imaging. So what does this have to do with chromatin organization? Actually, quite a bit. So if you think of histone deacetylase, HDAC, right, which plays a role in deciding how much hetero versus euchromatin you have, when your G-actin sort of polymerizes to form F-actin, F-actin is a substrate for HDAC, so it can come out of the nucleus. The other process also happens. There can be molecules that have substrates on G-actin that can go in. And in fact, these things reinforce this pathway and can lead to mechanical memory, etc. But here, this is the important process where HDAC sort of comes out. I'll show you data in a minute. And now what you can do is you can write down equations, etc. to figure out this reaction diffusion kind of process depending on how much contractility you have. And the bottom line is when you increase contractility, that is you put the cell on a stiff substrate, your HDAC actually comes out. The amount of HDAC in the nucleus is less. So the story that I'm going to tell you is when you change substrate stiffness, that changes actomyosin activity. And that leads to the shuttling of these epigenetic regulators. In other words, when you're on stiff, your HDAC goes out, right? And then when you're stiff, your chromatin is more open. When you're on soft, it is more condensed. And same thing with EZH2. We don't fully understand the EZH2 mechanism. We are working on it. Nuclear actin may be involved, so that's something we're working on. So bottom line, you look at this, this reaction or this reaction, either one. The soft environment will take you towards more condensed, and that's exactly what we see. And now comes the length scale question, right? What gives rise to these length scales? So with my background in sort of continuum mechanics, what we thought is let's take a really simple approach, where here we are seeing heterochromatin, euchromatin, and then the rest of what's there is nucleoplasm. So what we decided is we'll use a phase field modeling approach. The main idea here is that there are 3 phases. The amount of heterochromatin is CH, euchromatin is that, and the rest of it is nucleoplasm. They should all add up to 1. So it's a 2 variable problem because volume fractions add up to 1. And what do we do in these models? We include chromatin lamina interactions. We include chromatin-chromatin interactions. And more importantly, we look at acetylation reactions that can convert heterochromatin to euchromatin, and methylation that can do vice versa. So this will explicitly go in, and in fact it is these reactions that are going to give you the length scales. And now we want to look at dynamics. So we'll pick two, there are three variables, two independent variables, there's one constraint. So one of the quantities we'll pick is the amount of nucleoplasm, water. This is why I asked you what's the volume of the nucleus. Okay, that's actually quite important. I won't have time to show into this. You can actually squeeze the nucleus and see more heterochromatin. All right, so that's the amount of water, which is 1 minus the hetero plus U. And the other is a physics-based order parameter, which is the difference between heterochromatin and euchromatin. If it is a positive number, it's more heterochromatin. Negative number, it's euchromatin. And then what we do is we write down an energy landscape for this, which could come either from a polymer, continuum polymer theory with phase separation, or it could come from active matter physics, because each of these phases are being acted upon by by energy sources, and that can also give rise to phase separation. So what we assume is that there are two phases. And if you look at this phase, this phase has got a lot of water, and this number is negative, so that's more euchromatic. And this phase is heterochromatic. And now what we're going to do is we're going to write down the dynamics for the evolution of these two quantities and try to match that with what we see in our Histone experiments, both static and dynamic. So dynamics, okay, this bend it like Beckham, elasticity is important. I love velocity, that's what I've been trained in, but if you can put that back in, you'll get similar results, right? So, okay, now to the dynamics. So I have to write equations for two things, water transport and this order parameter, which is hetero to euchromatin difference. So water transport, there is more water in the compacted phase. So if this has to get converted to there has to be water transport. So details are not important, that is just the equations of poroelasticity. All right, next I have to write the equations for the difference between U and heterochromatin. Here, when you convert heterochromatin to euchromatin through this methylation acetylation reaction, that will change the difference. That's that term there, okay, very straightforward. This accounts for the fact that heterochromatin likes heterochromatin, euchromatin likes euchromatin, so they want to come together. So in other words, if you think about this is a conserved part of this evolution, this is the non-conserved part. And this is going to be the key part. This is going to give you all of what I'm going to tell you. So let's now look at this evolution. Sorry, one second here. So typically you'll be here. You're neither in this well or this well. Like if I don't put those reactions, what's going to, red is heterochromatin, the other is euchromatin. So what's going to happen is that you'll get phase separation. You'll get phase separation, and things will coarsen. You'll only get one domain, and the reason you're getting not a sphere but this thing sitting here is because of the fact that you have the wall. There's wetting there. That's why you're getting that. But now, if you put in the reactions, right, this additional reaction term I said, if you look here, you start getting characteristic scales, length scales. And also you get this wetting on the wall. I'll come to both of that in a second. So the question that we want to ask is, what is actually giving rise to these heterochromatic packing domains and their scales? So to look at that, so Yamini, there's a bottle of water there. Can you down there near my? All right, thanks, Yamini. All right. So the question is.
Wait, is this for an experiment?
What's that?
Oh, you want to drink?
Yeah. I just want to take a sip of water. Yeah, my mouth is dry.
Do you want me to fling this at you or something?
Thanks.
Need help from the audience here.
All right, very good. So next what I want to talk about is where do you get this length scale, right? So here, this where you are in this phase diagram depends on the methylation rate and the acetylation rate. High methylation, you'll get more heterochromatin. Low amounts, you'll get, and also water is important as well. So there you're in this part of the phase diagram. So you could have a homogeneous phase. But the homogeneous phase is not in these wells. So what you can do is you can nucleate a domain of heterochromatin, which is here in red, and then typically that nucleus will grow, right? And the nucleus is fed, the growth is fed by diffusion. But then this phase itself, because of the reactions, is unstable. So what's going to happen is that the diffusive flux is going to lead to the growth of this. the acetylation reaction is going to take it away, and this competition will actually pick a size scale. And what is the size scale? The size scale is, so the only variables you have here is a diffusion rate and a reaction rate. Diffusion is meter square per second. Reaction is meter per 1 over second. So meter square per second over 1 over second is meter square root is a length. So that's basically the length you're getting, okay? And of course it depends on what the far field concentration is. So it actually depends on the water content too. We sort of asking, in the first talk, when you squeeze it, you'll get more heterochromatin actually. The sizes actually will grow. We have data on that as well. And so what happens on a stiff substrate is you have less HDAC. So effectively the methylation rate is higher, right? The scaling you can do numerically and test. This is only an approximate analytical theory. And on a soft substrate, you get these bigger domains. But I didn't quite explain this. So to do that, all we also say is that every segment of DNA can interact with the nuclear lamina, and there is sort of an attractive interaction, okay? So the analogy here is you have water molecules in this room. On a cold day, water will condense on glass. That's because that's a denser phase. There's more, if you have attractive interactions, the phase that will form there is going to be the water or ice. Same logic here, right? So this becomes a surface wetting problem, and if you keep the methylation rate the same and increase the chromatin lamina affinity, you go from non-wetting to wetting. That is sort of from mercury, to water. And if you increase methylation, then you get these different, you get these bigger domains. Again, in the interest of time, when you go from soft to stiff, we can see what's going on there. Now, how do we test this? You put the cell on a stiff substrate and treat it with GSK or TSA. I'm only showing you the DSA data. And again, the sizes change. Yet another way of testing, we take the cell on a stiff substrate and treat it with a contractility inhibitor. So what we are saying is correct. It should look like soft. And it absolutely does. And Zizhan has a poster there. You can even extract the chromatin lamina interactions and things like that based on this sort of analysis. I will not go into it. I just want to, 2 minutes, right? So I just want to show the relevance to physiology. So we work with Rob Mock, who actually has cells from patients, from young patients with tendinosis. These typically tend to be athletes, long distance runners, etc. And from aged individuals, means 45 plus. And now, so you see the chromatin organization in these cells are very, very different. Now, what we did is we took these cells from the young individuals and put them on glass, stiff and soft, and cultured them and see how the organization was different. Turns out that on glass, it looks exactly like the aged, because aged tissue tends to be fibrotic, aged tissue tends to be stiff. The ones with tendinosis tends to be soft. So this looks like more like soft, and finally stiff is young. The last thing here is we can also look at dynamics. So what we can do is we can actually stiffen or soften these cells by shining light. These are hydrogels. So when you shine light, that induces cross-linking, it stiffens the gel. And then these are not, histome imaging is not real time. So these are not the same nuclei, but you fix these nuclei at different instances of time and see how the evolution takes place. You can very much see as you're going, as you're stiffening it, you have these smaller domains, etc. So you see that the domain diameter goes down, their number goes up, spacing comes down. So again, we can do the dynamics, but there's a very important bit here. You actually have to nucleate new domains. That's a very important piece here. That only when you nucleate these newer domains will they grow. And this movie will keep going until all of the nucleation is exhausted. So I'll just physically run this. At the end of the day, you get the phenomena we are talking about, all right? Finally, I don't have time at all, but we can actually put these reactions in standard polymer models and actually see the formation of these domains. They're working with Rachel McCord. You can treat the these 835 cells with TSA and look at where these the genomic changes. Actually, they all take place near the boundary. So the boundaries are a big way of are an important part of organizing chromatin, and all the changes take place within. You can read the numbers there, right? Okay. So with that, the conclusion is. Stiffness affects contractility, contractility affects HDAC, HDAC affects the way chromatin is organized, including HDAC also controls the nuclear lamina affinity. So that's the main picture here. And so we are now currently using high-C HDAC RNA-seq in my wet lab to look at how these things evolve with the stiffness of the microenvironment. With that, I want to thank all the funding agencies. And so we have we have Vivek. Everybody in red are working on projects related to this. So Vivek is here, Zhishan is here to around, he's here. All right, and also this work was started off by Xingyu Chen, the phase field modeling. So he's gainfully employed at TikTok, but with the new bill signed, I think he's moving to Facebook next week, but he's doing well. All right.
Thank you very much.
Over here. So what if we just go directly to contractility? I have two questions, so you can decide which one you'd rather answer. So
what if you
go directly to contractility and look at smooth muscle cells when they're exerting force or when they're not? Do you see these changes?
Actually, we're working with a colleague on smooth muscle cells, but we think we'll see it. We're actually seeing it in cardiomyocytes, which is not quite smooth muscle. So if you knock down the contractility in cardiomyocytes, then we see the soft phenotype. Go ahead.
So the other question is, what if we don't think about contractility, but you actually think about shape? So have you thought about looking at cells growing on patterned substrates like Matthew Peel makes? Because there you can have amazing different shapes of the cytoskeleton.
In fact, yesterday, I give this talk generally to mechanobiology folks. In fact, the third slide that you saw was actually a pattern one. That PNAS article, we have both stiffness and patterning. Stiffness and patterning are one and the same. If you make it elongated, it takes on the stiff phenotype. If you make a round, and also the cell area is also important. Both the cells on stiff substrate tend to spread a lot. So you can pattern it with the area and contractility, and you get the same sort of effects. Correct, thanks.
Okay, over here. I have a more general question. You see changes in the nuclear morphology, you see changes in lamina attachment, heterochromatin. What's the goal of this? Do cells use mechanics of does it change mechanics of the nucleus? And do cells use mechanics of the nucleus to adjust to the substrate? Like you were talking about going from the mechanics of the substrate to the events in the nucleus. The question is to what extent this changes the mechanics of the nucleus and whether cells might be using this for their...
Great question. So here I have some, that's a great question, some answers here. So here, here Wnt and ERK, or MAP kinase are very important for cells to metastasize. Okay, so these these genes are actually located quite close to the domain boundary. That's what we are finding. So when you go from stiff to soft, those genes are upregulated. So in other words, your change in chromatin organization will change to changes in gene expression, which can reinforce the stiff or soft phenotype.
Yeah, I was asking more about the mechanics of the nucleus, whether the nucleus becomes...
Oh, nuclear, okay, nuclear mechanics, yes. So roughly speaking, nuclear mechanics can be broken into two parts: the lamin mechanics and the mechanics of chromatin. So if you knock down laminase C, that changes mechanotransduction. You can actually change the way chromatin, that changes contractility. So all our things that we have looked at so far goes back to contractility. So anything that will change contractility is going to change chromatin organization. And knocking down laminase C will change contractility. It will affect that. Now, the DNA itself, right now, we don't have any evidence. We've only scratched the surface. More evidence may emerge to that effect.
What about lymphocytes that don't have laminase C?
Great, I didn't know that. Okay, so that's something we should be looking at. Yeah.
So if I go back to transcription regulation, you know better than I do that if you put stem cells, you get differentiation into different cell types.
Absolutely.
So,
Yeah.
So can you connect that in principle, or maybe are you working on connecting?
We are working on connecting. We're absolutely working on connecting. We're absolutely working on connecting it to differentiation and right. So the clutch sizes in these different the different phenotypes. So you take stem cell, it goes into a cardioma. They're different. They're very, very different, and we are absolutely working on. There's some data in Vinayak's poster there. We're working on connecting it. Yes.
Here, it's always very interesting when you put mechanics and chemistry acting together. And what I didn't get, or maybe I didn't pay enough attention, what are the time scales? When you're applying forces, how big the systems respond and change their acetylation?
Right. So again, this is based on our reading of the literature. So there are actually two processes: the reaction process and the diffusion. They all, if you work out, will turn out to be 10 minutes. But the changes that are occurring when you change the stiffness of the substrate, right? Stiffness of substrate is changing in 5 minutes. But chromatin organization is taking hours, 24 hours, 48 hours, at least in the experiments we've done. It may be bigger, the longer time process. So the thing is, we think nucleation can explain it, that the nucleation is a very slow process. And by analyzing that data, the nucleation rate is the rate limiting step for a lot of this evolution in the things we've looked at. That doesn't mean that over longer time scales, there may be a lot of other processes that may be reinforcing biological process. At the physics point of view, nucleation seems to be the rate limiting step. Right? Mechanics is fast, but you gotta get these newer nuclei of things coming together, and that can also impart memory. I mean, I didn't present this here. So we can actually cycle between stiff and soft, and we see memory.
And these cells we're looking at, again, they were myocard cells. What are they?
These are mesenchymal stem cells.
Okay, so don't they duplicate faster than the time to take to adapt then?
They duplicate in more 24 hours or more. So it's 24 to 48. So all of these things that I'm showing you is before they have duplicated.
But you say 10 hours, 12 hours, the thing get mixed up, right? The two things.
Yeah, you can. But we look at before they divide. There's quite a bit of evolution going on, between while they are in interface.
I have a quick question.
Yeah.
All these changes in chromatin compaction, do you know if they affect also proteins diffusion or even chromatin diffusion?
Affect what?
Proteins diffusion. If you were able to work in live cells, would you see a difference in proteins diffusion?
Protein diffusion?
Yes.
Which protein?
It could be histones, or it could be olaco, chromatin, or any other proteins.
So again, everybody here knows more than me. I think diffusion is going to be quite fast, I think diffusion probably can happen in minutes. Right, but this evolution is much slower. So you change the stiffness, it takes 10 hours, 12 hours to really adapt to that stiffness.
But if you compare in different substrate, like a stiff glass, and then your soft.
Right. So what we are doing is we're taking a soft substrate and hitting light. The light is cross-linking. It's making it stiff, and it cross-links it really fast. It cross-links it in less than 5 minutes. Mm-hmm. But chromatin evolution, even after 10 hours, is not done. Then, as Jose pointed out, they may divide, and things may become more complicated. Yeah.
Thank you.
Thanks.
Okay. Thanks again.
Okay. Yes, thank you. But generally what I'm going to talk about is what my lab specializes in single molecule measurements of biological systems, and one of the things we've been working on is studying nucleosome stability and chaperone activity. Okay, so you've seen this before. This is like the general idea of the length scales involved, and I'm going to focus on these very small, the nanometer scale, where it's just the nucleosomes and the organization of the nucleosomes, the wrapping of the DNA around the nucleosomes, and of course how much is wrapped determines sort of what happens at other length scales. But the most important thing is that base pair level transactions like transcription require destabilization of the DNA that's packaged in these nucleosomes. So I'm sure you all know, so histones coordinate to form a symmetric nucleosome. So you have a histone octamer. Of course, DNA is highly negatively charged. The histones are highly positively charged. You have about 150 base pairs wrapped around this octamer. For this talk, it's also important to know that the way that is assembled is the inner, we're calling the inner wrap, is the H3H4 tetramer. And then the H2A, H2B are kind of the outer wraps, so that when you disrupt the nucleosomes, the first thing that unwraps is the two outer halves of the first wrap, which disrupts the H2A, H2B dimer first, and then the H3, H4 is actually much more stable. So the question I want to answer is, or look into, is how stable are these wrap structures, and how can, of course, the RNA polymerase transcribe through the tightly wrapped DNA. To help answer this question, one of the things that we developed is a method where we put together an array of nucleosomes. So we don't want to just look at a single nucleosome. That's not realistic. Of course, this is an artificial construct, but it does have nucleosomes that can interact with each other. What we did was we set up so that we have 12 equally spaced arrays that are equally spaced because we use this Widom 601 sequence. Again, that's artificial, but it gives us the ability to quantitatively characterize when we disrupt these nucleosomes. And one of the things that we can do is after we assemble these nucleosomes, we also put handles on the ends. So if we assemble them correctly, you have 12 in the middle, and then you have sort of these handles. And that's because we're gonna catch these in optical tweezers and stretch them. But we took this same construct that we're going to use in the optical tweezers, and we put it down on an AFM surface. And you can see that we're able to assemble these nucleosome structures. You can see the handles at the end. And then if you zoom in, you can count actually 12 octamers. Occasionally you get random nucleosomes, but really on average we get 12 nucleosomes bound to the construct. And so the reason we did this is, and I'll get to a little bit more with the AFM, you can characterize with the AFM as well, but we're interested in how stable these structures are. And how do we figure out how stable they are? Well, we can actually disrupt the structures. And so this is what the experiment looks like. We have a polystyrene bead, or two polystyrene beads, one held in an optical trap, one on a glass micropipette. Sometimes there are two optical traps, you'll see that later. But in between these beads, these are micron-sized beads. We have this very short, which you can't really see here, a DNA construct with the nucleosomes. And as we then move the micropipette away from the bead that's in the trap, the bead in the trap moves slightly, and the distance that it moves is proportional to the force, and so we can measure piconewton forces on this construct. And actually, this has been pretty well characterized over the last decade or two, that you get these very nice force extension curves that are completely reproducible, especially for nucleosomes alone. So what we get is first you pull on it and you get. I think I have a diagram here. So when you pull up to a few piconewtons, you have intact nucleosomes until you get to a certain extension, and then you have a reversible, almost constant force phase transition where you're essentially pulling off the outer two halves. That's where the H2A, H2B dimers are being disrupted, and that happens in equilibrium, and so that means that if we stop in the middle and go back, it's a reversible process. What that tells us is that we're actually directly measuring the energy holding the nucleosome together at that level. However, once we've unwrapped the outer half wraps, we go to a much higher force, and then we get these sudden rips. And those sudden rips each represent completely disrupting the nucleosome, and that barrier is the barrier to unwrapping the tetramer, the H3H4 tetramer. And so each of these individual rips characterizes one of those nucleosomes completely unfolding, and we're showing them the nucleosome staying there, which we'll prove later on. So you can do this over and over again, and you get a pretty wide distribution of results. When you average those results for the inner turn, so I'm going to call this the inner turn results, you can measure the force, and you can actually fit it, so that's a non-equilibrium process, it's not reversible, but you can fit it to a simple model that basically tells you, if it's basically a two-state model where the nucleosomes are considered to be independent from each other, which works pretty well for nucleosomes alone. And if you fit it to that model, you can get things like the distance to the transition state and a natural rate of unwrapping, and as you might expect, this K0 is very low because the unwrapping rate, 5 times 10 to the minus 3 per second. That makes sense because nucleosomes are very stable, so their natural opening rate should be quite low. So what this tells you is that we have a way of measuring the stability of the nucleosomes, right? What we're really interested in is how are these disrupted, and how is transcription facilitated when you have these tightly wrapped nucleosomes. So we've been studying this protein called FACT. FACT stands for facilitates chromatin transcription. It's a non-ATP dependent histone chaperone. We use the term histone or nucleosome chaperone independently. Histone chaperone is, a lot of it is referring to these motor proteins that assemble the nucleosomes. That's not what we're talking about. We're talking about these non-ATP dependent, it's just a binding protein, and there are many of these proteins, and it's highly expressed in non-differentiated cells. And its FACT is a heterodimer. Cryo-EM images show it's binding directly to the histones directly to the histones, but this most recent structure that is available of this bound to a nucleosome did not have this HMGB domain, which we're going to show is actually quite important for its function. So what we wanted to do is to test how this nucleosome chaperone functions by looking at full-length FACT in individual domains on the stability of the nucleosomes that I've shown you that we can measure. And if you just look at our stretching curves of that same construct in the presence of FACT, what you see is that the nucleosomes are much easier to disrupt. They're much less stable. This makes sense. It's supposed to facilitate transcription. It should destabilize nucleosomes. And we actually took pieces of the domains, including the HMG domain. We had studied there are actually HMG proteins in cells that are just HMG domains or much smaller. And we've studied those in the past, and we see that they destabilize nucleosomes. So we decided to look at that, and indeed we do see that that also destabilizes nucleosomes. And so we do a full titration, and we get results basically showing that as a function, as you go to a high concentration where you have saturated binding, the average release force goes down for full-length FACT. It goes up for this MD domain that we know binds to the histone itself. And it goes down for FACT. So FACT destabilizes nucleosomes, and the HMG domain destabilizes nucleosomes. And one of the things that we might ask is, well, if HMG, this little domain, can do the destabilization, what's the point of having this whole huge protein? And we'll get to that. And let's see. So I wanted to also just sort of show that we can get a lot of quantitative information here. Again, here's the, this sitting to the two-state model for all the different domains. And what we see is that the opening rate increases tremendously. This would be the zero force opening rate. So, we're applying a force here. We're applying a force to sort of determine what the energetics are of the system. Although we know there are forces, and so forces can, can facilitate additional opening. And so we see huge increases in the opening rate for the MD domain, for the HMG domain, and for the full-length FACT. So overall, the HMG domain disrupts histone DNA interactions and enhances nucleosomal breathing at zero force. And finally, another experiment that we are doing uses an instrument that we acquired that allows us to both stretch these constructs and also visualize fluorescence at the same time. And so this can help us to answer that question about, we've disrupted these nucleosomes, but what happens to them after we disrupt them? And so we've labeled on H2, on the H2A, so all the H2As are labeled, so there are 2 per nucleosome, so we have 24 fluorescently labeled, 24 fluorescent markers. And we have the construct where we're stretching in between 2 beads. You can see the beads from autofluorescence, and you can see that when we stretch the DNA, the construct comes apart. And so when we stretch here in this curve, this is called a chymograph, we're following along this line as a function of time to see what happens as a function of time when we stretch. So when we move the beads apart and stretch it up to 40 piconewtons, that's a force where the whole thing has been disrupted. You've disrupted the H2A dimers, and you've also disrupted the inner structure where you get those ripping interactions. And what we see is that, in fact, the nucleosomes stay for minutes. Basically, the nucleosomes stay there. So even though we fully disrupted the construct, the nucleosomes are there, but there's no DNA wrapped around there. They're just basically stuck to the DNA, at least for that amount of time. But what we want to know also is then, let's say that that a motor has come through and disrupted the structure. What happens then, right? Can it reform? And so we notice that when we stretch and then release and we go back to 0 force and stretch again, depending on how long you wait, but typically we just wait, there's 10 seconds per cycle. You stretch again kind of right away, and you see how many of the nucleosomes reform. So when you go back, basically none of them completely reform, but you do see evidence of interactions starting to reassemble them. But in this case, you see after the 3rd cycle, we still have almost half the nucleosomes. So they do tend to reform even in the absence, for just a few cycles at least, in the absence of any binding protein. So this is, we call the survival probability. So the number of cycles the number of times you've disrupted the nucleosome, and then the probability that you still have a nucleosome that can reform. And then there's the release force, which tells you how stable it is. And then that's nucleosomes alone. And then we add the HMG protein, which is the one that destabilizes so that it lowers the force. And you see that it doesn't help to reassemble. It makes it worse. Right? So the HMG domain alone does not help to reassemble the nucleosomes. The MD domain, which is the part that binds to histones, seems to help a little bit. But again, it's not reassembling very well. However, if you add FACT, you get a much different story. It's both destabilizes the nucleosomes and helps to facilitate reassembly. So that's the reason that you have this whole big protein, right? You've got a piece of the domain, you've got a piece of the protein bound that you've got 2 parts, one bound to the DNA, one to the proteins, that helps to reassemble after you've disrupted the nucleosomes. Okay, so basically what happens is that the HMG and MD domains coordinate in the complete protein to facilitate assembly. All right, so that's actually published data at the very end of 2022, where we showed how these domains coordinate. And so the full FACT protein both destabilizes nucleosomes and strongly facilitates assembly. So FACT is the paradigm for non-enzymatic chaperone activity. Are there other similar chaperones? And there was this paper published in 2019 where the Reinberg lab went looking for such other examples. And the reason is that FACT is found only in abundance in non-differentiated cells. And so in this study, they looked and they found these 2 proteins, LEGF and HEGF, which are relatively abundant and expressed in most differentiated cells. And they found that they facilitated transcription in vitro, suggesting roles as nucleosome chaperones. To summarize what I said before about FACT, you have kind of this MD domain as part of this overall structure here that binds to the histones, and you have the HMG domain that binds to preferentially to bent DNA. I didn't mention that before, but we know that's the case. And LEGF and HEGF have something similar. So they have AT hooks, which are like HMGA proteins, which are kind of like the HMGB domain that we have here, in that they bind bent DNA. And they also have this PWWP domain, which binds to modified to methylated histones as specific methylation. And so the hypothesis is that this might function in a very similar way. Even though it's not a heterodimer, it's a monomer, it may function as a homodimer. And so we wanted to test the biophysics of this to see if this might be, in fact, the nucleosome chaperone for differentiated cells, and how does it work if it is. And so, you can get these curves, you can add, we have the modified histones, which I'll talk about in a minute. We have HEGF, and then we also have LEGF, and what you see is there are definitely effects both on the modification on the stability of the nucleosomes and of the protein on what we see in our force extension curves. But they're messy, and you have to do many measurements to try to figure out what's happening. So the first thing is the methylated histones. So these domains bind to H3K36Me2 and 3. We have H3K36Me3 nucleosomes that we've assembled. They're a methylation mimic. They're not native, but they're believed to be very, very similar. And we'll see in our experiments if they bind that domain. And we see there is an effect, but if you look at the outer wrap, you see, within uncertainty, very little effect on the stability, and same with the inner wrap as well. So they don't have much. We get slightly different results, but we don't have much of an effect on the overall average stability of the nucleosomes. If we then measure this release force of the outer wrap, again, this is a few piconewtons of force. What we see is that for unmodified nucleosomes, there's a little bit of an effect of LegF on the stability, and a little bit of effect of HEGF as well on the outer wrap, but the inner wrap is very stable. Now, what happens if we combine this methylated nucleosomes and these proteins? What we see is that there's a huge effect of LegF on the stability of the outer wrap. Essentially, it's unstable. And even the inner wrap is slightly destabilized for LegF. So this suggests this really is destabilizing. And not only is it really destabilizing the nucleosomes, but it is specifically destabilizing methylated nucleosomes with this one particular methylation modification. And I tried to, we're still working on this, and the literature on these modifications and what they do. I found this paper which said, they're associated with the transcription of active euchromatin, consistent with the role, which is consistent with the role in facilitating transcription. But I think there's a lot of disagreement, and that was saying, well, some studies show this and some studies show that. So I'm not going to have any conclusions about that, just to say what we observe is that this modification in the presence of these proteins does appear to destabilize nucleosomes. We also observe the same thing when we look at the spread of these constructs in AFM, that specifically that methylation gives you further unwrapping of the DNA, showing that, and in this case, both of these proteins cause additional unwrapping of the nucleosomes. I know I'm out of time. The kinetics are changed by the presence of LegF and HEGF. They actually dissociate more easily in the presence of these proteins. That's consistent with fact. Basically, the idea is that it increases the kinetics of the interactions. Okay, so finally, histone and DNA binding domains of FACT coordinate to both destabilize nucleosomes and facilitate reassembly in undifferentiated cells. LegF and HEGF are found in differentiated cells, contain both histone and DNA binding domains. LegF and HEGF destabilize nucleosomes. LegF strongly preferentially targets nucleosomes containing the H3K36Me3 histones for destabilization, at least in our experiments with the methylation mimics. These modifications are, according to that paper, associated with transcription of active chromatin. And the results suggest LegF and possibly HEGF are nucleosome chaperones for differentiated cells regulated by the H3K36Me3 modification. And finally, just acknowledgments. Mike and Macaulay did all the work on FACT and worked with graduate student Joha Joshi on LegF and HEGF. And Micah has a poster actually here on nucleosome acylation and its effects on stability. And Joha has a poster on the results I've been talking about with LEGF and HEDGEF. And this work is a huge collaboration. Yulia Razuna came up with the ideas, working with the Maher Lab at Mayo Medical School. Carolyn Luber provided us with the histones, especially the fluorescently labeled ones. Dmitry Lyumkus is our direct collaborator on studying, and he provided it with the methylated nucleosomes and the HEDGEF and LEGF proteins. So thank you.
Mark, that was absolutely just gorgeous. The whole, the way that you've managed to get these beautiful 12-mers and analyze them in parallel is really very gorgeous. So I'm curious, given the obvious sort of puzzling finding that when the chromatin fibers folded, so you accept a clutch's 8 to 12 nucleosomes, right? There's going to be some high order structure, which you can see by sedimentation analysis. Jeff Hansen showed this years ago. The H4 of one nucleosome interacts with the H2B of the next, makes a very stably folded fiber. But how come when you do the tweezer experiments that folding doesn't seem to be particularly resilient? And what's the implication of that when a polymerase is chugging through? Does that mean that the polymerase doesn't care, doesn't see higher order folding, and it really only cares about that single nucleosome that it's encountering? And how do you square that with the Carlos Bustamante papers, where they do see some role for high order loop folding in the way the polymerase transits through? And I'm curious if you can address that with the modifications. Thanks.
Right. So the higher order, you mean folding beyond the nucleosome wrapping?
Right. I'm talking about folding into one or maybe two loops, or maybe even a crisscross model if you go with Sergey and Chris Woodcock's ideas.
Yeah, I mean, we can't address that really in this experiment, right? So I don't know what the.
Well, so one idea would be if you put H1 in, onto the optical array, onto the array that's for the tweezers, do you see a fundamental change?
That's a good, there was a discussion of H1, right? So we haven't done anything with H1, mainly because it makes our experiments messy, right? Because it sort of, condenses this whole thing. And it probably competes, one of the ideas was that it competes with these nucleosome chaperone proteins. And that might be the case, that there's an interplay between those. We haven't done the experiment, but we could. And then of course, then there's that question, which H1 do we study? But indeed, that can play a role. I mean, we're just assembling kind of the simplest system that we can go with to get quantitative measurements out. But there's no question that further complex, as the system gets more complicated, as we add more to it, there are going to be more interesting effects. And indeed, I'm sure that there are additional effects. One of the things, in our construct, the nucleosomes can't stack on each other, which that they have to normally. And they can't stack on each other because there's a rigid linker between them. But as soon as you add anything that has these HMG domains, it bends the DNA, they start to stack, right? So, all these different effects are going to come together to give you a huge interplay of different effects. So I agree that this is just a simple model, and we need to build complexity into the system to learn more about what these proteins will do when they encounter other barriers. Yeah.
No, here, thanks. So I was wondering, when you are extending, applying force and disrupting nucleosome, but still having histone bound, as in the picture here, do you reach the length that you would expect for totally naked DNA, or do you think that you still have some kind of little compaction provided by the histones that are staying bound?
You mean when we fully stretch it, towards at the very end? Is there? That's basically the stretch. No, that's basically the stretching curve of double-stranded DNA. So it's bound. It's got to be bound, right? And there's charges there, and there's some interaction there. So if we had really good signal to noise, there must be some effect. Right, it's just that when you average over the polymer properties, we basically don't see a significant change in extension.
But do you think that it stays as an octamer? I mean, you are
right. Do they stay at the same place? Right? Is that we think they mostly stay at the same place, but that's a question that the fluorescence can try to answer. And there is, with these binding proteins, we think that it also can lead to some sliding. So one of the points of using the 601 is that it localizes it, right? And so in a way, it's not a natural situation, right? We're already localizing it too much. So if it can slide when the 601 is there, it's definitely going to be sliding all over the place when it's a different kind of site. So I think, and we do see that with the chaperone proteins there, they slide more. So I think there is some effect of those proteins in making it easier for them to slide. But again, it's an artificial system.
Thank you.
This is actually potentially embarrassing, but would it be useful to do an experiment where you could increase the force, then have it drop back, increase? So I'm looking at your reproducibility, because there is a cool way that you could do that, which would be putting something like a piece of titan as a linker. Because as you stretch that, then you unfold IG domain by IG. So you would get, and then you would drop back, up and back. What would you expect you would see if you could do that?
Yeah, I mean, in the outer wrap disruption, right, that is reversible already, right? So, if you go back, it's already going to reform. I think it is an irreversible process in the sense that if you go back right away, you have some small probability that it will reform. And there are theories that describe exactly what that probability is. You can do something where you sit at the you use feedback and you sit at the force. So as soon as it goes down, you move it back, and it's like a constant force feedback. And people do that, and you can kind of hold it in a sort of an equilibrium and try to get a measure of how stable it is. Yeah.
So can you use any form of cross-linking to look at when you've got them unfolded and they're still stuck to the DNA, how reproducible is the surface of the unfolded octamer or whatever it is that's stuck to the DNA? Is it always stuck in exactly the same way? Could you look at that by cross-linking?
Yeah, I mean, because it's a single molecule, we can't recover that molecule. And, so you would have to have something fluorescent that you have to do it with some kind of a fluorescent marker, I think. But that kind of experiment should be possible. It's all about whether we have the resolution and do we know what we're really looking for. If we have a very specific hypothesis, I think that could be done. Yeah.
I think this is fantastic because it's basically all this discussion. I just want to go a little bit further on that. When you say disrupt, you always disrupt the same way. What's the distribution of times that the nucleosomes stay there, right? Basically, histones do not move. Do you have a distribution of times, and does it depend on the DNA sequence where it gets stuck?
Right, so we don't know which one is being disrupted. Even in the fluorescence experiments, we're just seeing, all the fluorescence. We know only the number. And basically the whole theory is the theory that we're fitting it to says that the force at which it disrupts goes up by, on a log scale. And the reason is because you're applying at some force, and then there's some probability of it fluctuating open. And if there are 12 of them to fluctuate open, there's a higher probability of one of them fluctuating open. So I don't think I think it's probably random, but we don't really know, right? It probably could be any one of those 12. I don't think, it's a uniform force throughout the molecule. We're just providing, we're basically lowering the barrier to all of them unfolding. And so whenever one unfolds, that's what we see.
Right, yeah. But if you repeat the experiments, you get always the same time as you disrupt the same force, the time that takes to move away is about the same, or there's a distribution?
No, there's a pretty big distribution of the different results. I showed that, you see that one nice curve and you see those rips, but then when you have the plot, I don't know, it might be easier to, so here. So this is, you can see how many remaining nucleosomes, and the force distribution is huge. So I don't know about the timing maybe, but the distribution is huge, so I'm sure that the time distribution is well, yeah.
That's pretty cool. Thank you.
Thank you very much.
Thank you.
So let's thank Mark again. So now we're...
Yes? Okay. Thank you very much for the invitation to come to Boston. It's one of my favorite cities, and this is a very intimidating audience. I should preface my talk by saying I have a confession to make. My name is Yamini, and I'm a biochemist, not a biophysicist. So I'm looking forward to getting feedback from all of you on all the things, all the models that I've gotten wrong in my head. So my lab has been interested for a long time, since the beginning of graduate school actually, in this absolutely fascinating problem, which is that eukaryotic DNA is not naked in the nucleus. As you heard from Bill, the density of nucleosomes is extremely high in the eukaryotic nucleus. It's 1 mg per mil. If you just burst open a nucleus and touch it, it's like this sticky, snotty glue. But of course, eukaryotic bacteria do it very elegantly, right? So you've got basically this gorgeous operon unit. You know exactly where the transcription start site is. You know the polymerases are going to be. There's transcription factor-driven dynamics mostly. But here is a really beautiful example of Drosophila chromatin. And what you have is this beads on a string, and then you've got these large transcriptional complexes occurring, all in the context of nucleosomes. We know a lot about nucleosomes. They've been studied for an embarrassingly long time, considering how little we know about how they work. And of course, as you can imagine, this beautiful octamer that Mark referred to earlier, very beautiful work from Mark's lab, you're basically wrapping 147 base pairs left-handed around the nucleosome, and you can make all these beautiful three-dimensional structures, which are not actually as regular as I show here in this review, because the linker lengths are different, the histone modifications are different, the spacing is different, and of course every now and then a nucleosome will just pop off and disappear and come back on. So it's very dynamic, and these structures are probably interchangeable. So the question is, are nucleosomes a hindrance or an opportunity? And I think that depends on your perspective. If you're looking from the chromosome down, like Bill, maybe nucleosomes are a hindrance. If you're looking from the beads up, you see them as an opportunity. And then the question is the genome a self-organized polymer or not? And of course, beautiful work from Job and Leonard and other people has contributed to this debate greatly. I love to show this because this is the reason I fell in love with chromatin when I was an undergraduate in Bombay. This was the first micrograph ever taken of chromatin. This is actually from the Oldensies, but Chris Woodcock has even just as beautiful images. And what you can see is this really gorgeous beads on a string motif, but somewhere in here is a gene. And somewhere else in here, this polymerase has to find this gene, go here, turn it on. Right now my adrenaline's really high, so it's probably pumping out adrenaline. But then there's something else that maybe you don't want to turn on, and you want to bind it, you want to close it off. And so the polymerase shouldn't go there at the wrong time. If it does, then you're going to be in trouble. So there's this interconversion between open and closed states, and the question is, how does that happen in the context of millions of these fibers close packed inside a nucleus? So it turns out there are lots of histone variants. It's not just a standard structure of 1H3. There's actually variants, 2AZ, and these variants have been shown to be associated with various chromatin states. The one I'm going to focus on right now is one Bill discovered. It's called SEMPE, and it's a great model for understanding epigenetics. And so somebody defined epigenetics earlier today as inheritance of traits. That's true, but in the common parlance, we now use it as whatever controls things above the DNA that's not essentially a transcription factor. I love this video from Ted Salmon's lab, and it shows you very beautifully the process of mitosis. Here you can see very beautifully the chromosomes in red, microtubules in green, and in there is a locus, and that locus is bound to the protein that Bill discovered when he was Hopkins from patients actually that had scleroderma, and Bill found antibodies that reacted with the centromeric proteins A, B, and C. These proteins define the inner centromere. Somewhere in here is chromatin, and you can see the microtubules bury in this outer kinetic core. The inner kinetic core is chromatin composed of these centromeric nucleosomes. This is an essential protein. It forms the basis of the whole centromeric complex, which is like several, megadalton complex. There's an array of SEMPE nucleosomes. There's an H3 variant, binds to a SEMPC. If you knock out, this is a beautiful paper from Andy Chu's lab. If you have SEMPE or SEMPC or a happy little mouse, you're eating cheese, If you knock either of them out, you're an unhappy mouse, you don't eat any cheese, and you just die. So that's very sad, and you obviously want to maintain this complex at the right place at the right time. So you would predict, right? SEMPE goes to a particular DNA sequence. It likes something specific, so it knows exactly where to go at the right time. Well, that turns out not to be true. So this is an astounding result. Here you can see H3, it's invariant. Like barely 1 or 2 residues are going to be different. You could be a P, you could be Donald Trump, you could be Joe Biden, you could be me. And no matter how we look like from the outside, how we behave on TV, we generally have really the same chromatin when it comes to H3. When we come to SEMPE, really there's a huge amount of variation. This is called the centromere paradox. In all of these species, you can knock out SEMPE for the most part, except butterflies, and you're dead. But the protein has very little in common with other SEMPEs. Furthermore, we know this from beautiful work from Glenys Logston using nanopost sequencing. This paper just came out this year. Even amongst us in this room, our centromeric DNA is fast evolving. So it turns out we don't have the same repeat structure. So how can you have a conserved process when your centromeres are not conserved? This is called the centromere paradox. Another way to ask this question is, so what is special about histone variants? So there are 2 possible explanations. One is they provide some kind of mechanical feature. They have something in them that defines their locus. So if you want to be transcriptionally active, you're very floppy or loose, and you have like this little handle, polymerase recognizes you, comes along. Alternatively, it's epigenetic, so you're purely a signaling molecule, so you have no really structural state that matters. It's just the signal that you have on you. And so for those of you who don't want to hear the rest of the talk, the answer is really both. So I've been interested in this idea for a long time, and this is a review I wrote as an introduction to the centromere field when I was joining the centromere field, in which I was sort of summarizing all the papers I had read from lots and lots of wonderful people. Most of the papers were from Bill, but Robin Alshire had a bunch of really great papers. And the idea was that maybe centromeres have something in them that helps them pop out. And maybe this something that pops out, helps them pop out, helps them get recognized very quickly as you go from prophase, all the way out through to anaphase, so that the kinetochore can bind. And I've been obsessed with this model for a very long time because I like this idea of like bunginess. When you look at the microtubules binding to the centromere, it looks very physical, right? You can see like the stretching and the pulling, you can imagine it. And so this was sort of like, has been obsessing me for a long time. And we started out by doing all-atom computational modeling with Garrick Papone's lab, who's a leader in this area. This is the grad student who did all the work a few years ago. Sorry, there's a problem with the, here, okay. So I'm going to show you these videos very quickly, if I can play them. And what you can see here, we're just going to look at the SEMPE nucleosome. There's a scissoring motion right here at the heart of the nucleosome core, where the residues are the most different. And H3 actually doesn't do that. It moves in a coordinated fashion. And if you look at the energy basins, the free energy basins that these nucleosomes occupy, SEMPE looks like it has the ability to tumble between different conformational states. I was really excited about this, and I said, well, okay, how do we test this sort of idea? We'd been doing AFM work on purifying SEMPE from human cells. We could see that it was occupying what looked like different shapes over the cell cycle, but we didn't know if that was just instability, perhaps chromatin modeling. It was really hard to know that. So Tatini Rakshit joined the lab. She was an AFM expert. She's now in her own lab back in India doing very beautiful work, along with Mary Pittman and Daniel Melters, who's a staff scientist. This trio came up with a way to actually do nanoindentation microscopy. So this is using AFM, but now instead of observing things, we're actually going to touch individual nucleosomes. And for somebody who's been obsessed with nucleosomes my whole career, I can't tell you how exciting this was when we started doing these experiments. We use standard salt dialysis. We make the chromatin. We always have magnesium and salt in actual buffers. So these are not dry. This is under liquid. And now we can use the AFM tip. We use a 10 nanometer tip to gently push on the nucleosome. It bounces back. We can do this, like 25 times per nucleosome to get the memory state that Bill talked about. And from this value right here, under controlled force, we can calculate the Young's modulus of each nucleosome. This had never been done before, and we got a big surprise. Here's H3 in fluid. These are each of them is a little nucleosome. You see the DNA in the back. Here's the centromeric protein from humans, and this is the centromeric protein bound to that closest kinetical partner, SEMC. Both of these are essential. So first, what you see is H3 is pretty stiff. It's about 12 megapascals. SEMPE is very soft. It's about half of that, so it's actually pretty elastic. But here's the cool thing. You start adding SEMC, and to a certain extent, you essentially rigidify that nucleosome so it kind of swells up and gets like tough. So you can actually titrate. This has never been demonstrated before for any nucleosome ever. that you can take a nucleosome, it's inherent, there's nothing else in here but DNA and histones, there's nothing else, just salt. And that's sufficient for this nucleosome to start out being inherently elastic. It's encoded inside its own protein sequence. And then when you add this kinetico partner, this is a very short fragment. It's only 12 amino acids of SEM-C that binds the C terminus, and that is sufficient to stiffen up this nucleosome. We did a lot of controls, so I won't go through all of that here, but that work is published in PNAS, and Jose was actually the editor for that paper, so thank you, Jose. Very supportive. What we were trying to figure out is what this meant for the biology, because this is what we care about in our lab, is how does the SEMPE function of mitosis rely on this? So it turns out nucleosomes are not at all static. I know we always model them as such, but they're actually constantly moving. They can slide, they can bop, they can jump. The whole fiber can fold and unfold, and all of this is happening in real time within the order of seconds. So if SEMPE is elastic, well, what happens to the mobility of those nucleosomes? Does it become more mobile, less mobile? Does it leap around? Does it try to jump? Does it try to expand the space that it takes, as I had originally sort of hypothesized? And then in the presence of SEM-C, does the opposite happen? So it turns out there was no really way to answer that question, so we decided to come up with a way. And this is all work from Daniel Melters, who's a staff scientist and who says hi to Job Decker, because apparently we're collaborating, which I didn't know, but it's cool. And Keir Newman, who is a single molecule expert at our sister institute, NHLBI. So Keir and Daniel decided we were going to do this quantitatively. And so we came up with a way to, as I said, we reconstitute chromatin all the time. We could do it in our sleep. We use high-speed atomic force microscopy, which is an advance, where you just scan very, very, very fast, very gently and very fast in buffer, and you can actually see nucleosomes moving in real time. And again, for those of us who love nucleosomes, this is cool. So here is a single SEMPE nucleosome. You can see actually it is deforming. It can, 2A and 2B will split off and come back on. It's sliding around. You can assign beats to each nucleosome, and you can actually track this over time. This is real time, 18 seconds or so. And then we can do this with SEM-C. We can even do it with H1, but I won't show that data today. All the H1s, Bill, 7 of them. And you can essentially capture these trajectories over time. We want to do this quantitatively, and so this is where Curie really helped us using single molecule methodology. We analyzed 6,000 nucleosomes. It's never been done before, probably will never get done again. But we wanted to set a bar for the whole field because people were really reporting 2, 3 mononucleosomes, which is like rubbish, basically. So we wanted to do this right. They came up with a single particle tracking approach, drift correction. Only 3,000 of these 6,000 nucleosomes pass our quality control. And from all of these data, here are the qualitative videos. What we determined is that SEM-C restricts the motion, the freedom of motion of SEMPE. And see, these are the individual trajectories over time, and this is what happens when you bind this particle. H3 is not at all as mobile as this. Of course, we can quantitate this using MSD and by looking at diffusion constants, and in both cases, SEMPE nucleosomes are actually very diffusive, they're very mobile, and SEM-C in both cases will completely halt and slow down that motion. And this is up to a certain point. You can hit a plateau with SEM-C. If you add too much SEM-C, everything just gets gummed up, so you have to be very careful. So we're like, okay, what does this mean for the cell? And how do we actually test this idea inside a living cell? So we did a very obvious genetic experiment, which is we'll just overexpress SEM-C. It turns out that in nature, SEM-C is never overexpressed. SEMPE is in cancer cells. We discovered that. Shenoy Balmuzni and I published papers on this when 10 years ago now. But it turns out in normal cells, the amount of SEM-C is highly regulated. And we were wondering what that meant. So we said, okay, what if we just overexpress SEM-C? And presumably, if the same thing is happening in vivo, which we haven't formally demonstrated, you might expect there's an impact on SEMPE openness or the accessibility of the SEMPE chromatin. We looked at RNA pol2 on the centromeric fiber. We and others have seen that polymerase is on the centromeric fiber in living human cells and is required to load new SEMPE. If you overexpress SEM-C, what you see is the amount of RNA pol2 drops. These are just 2 different antibodies. This is the antibody that Bill discovered many years ago that stains the centromere. And if you look then at the amount of SEMPE actually begins to go down, and RNA transcription from the centromere goes down as well, which we had shown was required for SEMPE loading. We can look at new SEMPE, which is de novo SEMPE epigenetic renewal, using a quench pulse approach developed by Lars Janssen in Don Cleveland's lab. And when we do that, what we see is in the overexpression of SEM-C, new SEMPE loading is inhibited. And what this means, I think, this is our model, it's a working model, is that in the presence of SEMPE alone, the centromeric transcription, the fiber can be transcribed by polymerase. It's completely accessible, which is intuitive. You have a nucleosome that's very elastic, it's very mobile, it makes sense that polymerase can invade that fiber. If you want to protect that fiber and you want to shut it down, you can load a protein called SEMC, which presumably protects that fiber from being invaded and disrupted. And this is presumably important during mitosis. So this was an informal sort of deduction, right? So we don't have evidence in vivo what SEMC is doing. So we want to test this directly. It turns out we can push boundaries by AFM. We're doing this all the time. We really love it. We love doing this kind of stuff. So this is high-speed AFM, as I showed you, just nucleosomes bopping around. The question is, can we add transcription factors? Can we add polymerases? Can we observe the act of transcription? So Daniel has been working really hard on this, and I don't have the actual data yet to show you, but the idea is, can we add polymerases? Can we add chromatin remodelers? So I'm going to show you his first really beautiful success, which is just taking naked DNA to which we're adding T7 DNA polymerase. And you're watching this over real time. What you can see is the polymerase is already generating supercoils as it transits along the DNA fiber. So this is very exciting because the next step should be very easy for us, which is to put nucleosomes on it and then try T7 and eventually RNA-Pol2. But the goal is to then ask the question, does the histone variant impact the rate of polymerase transgression? Does linker histone H1 make a difference or not? Does high order folding make a difference or not? You can do all of these things. So that's the next advance that we hope to be working on. But the working model is the epigenetic, the mechanical state of these nucleosomes, we propose, or we hypothesize, is directly reflective of what is occurring to them epigenetically. So you have a mobile fiber with elastic, bouncy nucleosome. I feel like this might be hard to repress. It might take up more volume inside the nucleus. But when you want it really compacted and ready for mitosis with the kinetical complex bound, then I think you need this three-dimensionally folded state. So I'm going to end very quickly. I don't know how many minutes I have left.
5 more minutes.
Oh, good, okay. So I want to talk about evolution, right? So I pointed out the thing that really frustrated me about centromeres. Steve Hanikoff came to give a talk when I was a grad student at Purdue. And this was the slide he showed, which was that H3 does this, but SEMPE does this. This was Harmeet Malik's work. And this induced me to go work for Steve, even though I found him very frightening. Because the question was, well, what is the purpose of all this variation? Like, why do we have it? And so one idea I had is, well, if SEMPE and centromeric DNAs are both fast evolving, then neither alone is sufficient to say this is the centromere. So there must be something else. And that something else might actually be the mechanical state. So maybe being floppy has nothing to do with transcription or anything else, but it's a product of trying to escape repetitive DNA elements that invade centromeres. The reason centromeres are full of what is called junk DNA is because transposons really love to jump into the centromere. The reason they like that is because it's a safe place to go. You're going to get inherited to the next cell cycle. So that's true. Maybe SEMPE is evolving away from preferring a particular DNA sequence, in which case, if we look at SEMPEs across the species, we might actually get some clues as to when and why elasticity or even high mobility evolved. So this is a magnum opus project by a new postdoc in the lab from Arizona State University, who's an AFM expert. What Judith has done is we've cloned all the SEMPEs from all the species we could think of, covering the tree of life, and we've had them made into proteins, and she's reconstituting each one by one and doing elasticity measurements, eventually high-speed AFM. 6 down, 10 to go, and I thought this would make Bill happy, because it's really cool to think about evolution of centromeres. How do we get to where we are today? So SEMPE, this is the yeast centromeric protein, this is the plant SEMPE, and these are 2 mutants that are found in glioblastoma, which is a separate project we have in the lab. What Judith is finding is that it's not so simple. H3, as I said, is stiff. H3.3 is actually pretty stiff still. H3K36, which I think Mark just talked about, is a little bit stiffer even. SEMPE is soft and elastic. Yeast SEMPE is actually pretty stiff. It's almost as stiff as H3. And plant SEMPE is weirdly even stiffer than that, and we don't actually know what that means yet. So the idea is now to do high-speed FM and to see if elasticity is a predictor of mobility. Are the 2 properties related, or are they completely independent? And if they're related, does it affect transcriptional access? So stay tuned for that. I'll end with this. Part of the epigenetics is being at the right place at the right time with the right chaperone. And Mark beautifully, just gave a great example of why chaperones matter. As I pointed out, if histones go to the wrong place, with the wrong people, at the wrong time, bad things can happen. We and others have worked a lot on SEMPE in tumors. It's a biomarker for many cancers now. And we showed many years ago that SEMPE in colon cancer, for example, is present not just at the centromere, but outside the centromere. And Genevieve Almuzni has done beautiful work on this, as have many other labs. And we and Genevieve both discovered that SEMPE is essentially being hijacked or is hijacking. We don't know which. It's using a transcriptionally coupled chaperone pathway of H3.3 to enrich at ectopic loci. So this becomes like a really great model for us to test this idea of mechanics, right? Because if SEMPE is going to big domains outside the centromere, we have a new handle for looking at the change in this domain, this mechanical state, if it really matters. When you put a new histone in a place where it doesn't belong, and there are hundreds of SEMPE nucleosomes here at this particular locus, and it's recruited by an RNA. So you have like millions of these chromosomes that have the wrong locus here. Doesn't matter if you change that big swath of chromatin. And so we can actually ask this question because we've adapted our AFM method for nucleosomes, chromatin, chromosomes, nuclei, cells, and now even 3D bioprinted tumors. And so we've been able to apply this technique for whole chromosomes, and I won't show you all the data. But inspired greatly by work from John Marco, who I met at a conference like 4 or 5 years ago, and I felt like we should do this. So this is metaphase chromosome spreads in buffer. That's why they look a bit fuzzy. You can see this with the light microscope vision of the AFM. This is AFM mode for the same thing. We can do elasticity mapping of the entire chromosome set, like hundreds of metaphase spreads. And then Arun Ganesan, who he was in the lab and now has his own lab at the University of New Mexico, would run back with the same slides that were unfixed, fix them, and do IF-FISH for specific loci. So in this case, we're looking at chromosome 8, which has a SEMPE domain outside the centromere. And then we want to do high throughput pipeline for metaphase spreads, overlaying the FISH data with the AFM data. So the first part of this has just been accomplished by our high-tech core facility. We take thousands of chromosomes. We can use a spot detection algorithm that can detect any IF-FISH spot for hundreds and hundreds of such loci. And this is the workflow. If you're interested, we should hopefully release this protocol to the field in October. And then you have to identify, segment individual chromosomes, which we can now do. This is actually, the algorithm is doing it automatically, so we don't have to do this by hand. And then finally overlay the AFM data to find out whether a specific locus, when it takes on a new mechanical signature, is more likely to break or not. I'll end with this idea: why does this matter? So we published this paper with Payal Sen, who is a new investigator at the National Institute of Aging last year, and we followed up with our own work, which is hopefully going to be accepted at some point this year. But we looked at what happens to chromatin when it ages, and the biggest thing that happens to chromatin when it ages is heterochromatin is completely messed up. There's a massive redistribution of heterochromatic chromatin marks across different organs in mouse. We did some of the sort of similar work in aging humans, not me actually, but cells from different humans. And we focused on the centromere because we're really excited about looking at what happens. So this is just a metaphase spread of SEMPE staining, SEMP-C staining in metaphase spreads from a young. And now we've done this for many different age groups. And at the time I was 47, so aged. And it was not good news, like really didn't look so great. And I was feeling quite sad about myself that day because I was like, well, already at 47, if this is our state, I can only imagine what happens when you're a little bit older. And maybe a hypothesis, becoming a PI ages your centromeres, I'm not sure. But nonetheless, what we can do is take this idea of this overlay detection program and now purify aging chromosomes or purify chromosomes from cancer cells and ask the obvious question: is this weakened centromere something to worry about? Does the mechanical state matter for the function of the centromere and for ectopic loci? I'll end with this idea because Bill raised it in the beginning, so I'm kind of glad he did. What's the state of a centromere, right? What's the state of chromatin? We don't know. Like, is it a gel? Is it a solid? Like, what is it? It's very, very dense. We know that. So one idea we've had from the centromere work we've been doing is that we've been thinking that maybe it's kind of like a gel because there's not just nucleosomes there, there's also RNA. And when you have this RNA, it seems to be required because we see it still hangs around, it does not disappear. There's old data that if you get rid of the RNA, the whole centromere explodes. This is Conley Ryder's work. And we haven't quite done it in the way Conley did, but one idea is that you have this clustering. And just like when you pour a page gel, the amount of bisaccharidamide, which is a cross-linker, changes the stiffness of the gel. I have sort of this hypothesis that maybe RNA sort of does the same thing. So you have too much RNA, it's like Goldilocks, bad, you're dead. Too little RNA, like in aging cells, also bad. And then maybe that doesn't allow this domain to be as rigid as it should be for microtubule binding. And we can test this model by using some of the protocols and techniques that I showed you today. So the idea is that you have dysfunction in aging cells because centromeric RNA and SEMPE are both low. And if you restore them, which we can do by using LSD1 and P53, then perhaps you can re-resurrect sort of this gelatinized state or the solidity of the gelatinized state. This is just a hypothesis. We don't really have any good evidence for it, but it's one that we're sort of working to develop. With that, I would be very glad to take any questions. These are the people in the lab. Arun's now in his own lab. Shweta is actually on the job market. She's out at the Gordon Conference giving a talk on her work at just an hour away from here. And Judith is doing all the AFM work. And Daniel Melters is our high-speed AFM expert. With that, I thank you for your time. Be happy to take any questions. Alexandra.
Thank you, Yamini, for a beautiful talk. I was wondering, when you do your squishing experiments of the nucleosomes, if in addition to the elastic recoil, if you have looked also at the viscous relaxation, if there is some of the slower response. And a second part to that question would be, did you do your tapping also in a frequency-dependent way, so to obtain the whole complex viscoelastic modulus?
Sorry, what was the frequency-dependent manner? You mean like recurrently?
The tapping, if you have tried to basically obtain your
Young's modulus?
Exactly, as a function. Well, you focus on the elastic part, but if you were to go for viscous and elastic part, right, so to look at the viscoelasticity, so you would do the measurement in a frequency-dependent way of your...
That's a good point. We haven't done the frequency-dependent manner, but it's a good idea. We could certainly try it. We do typically look at both the repressive and the expansion, right? So both of those go into the force modulus in the Young's modulus equation, which is modeled using the AFM software.
And in the elastic recall, do they actually recover the original
Yeah, no,
size,
you can actually press them at least. So we press them 25 times typically because you want to capture all the space, because you can't exactly put the tip exactly where you want it. So typically you want to press, you go up, you do it again and again and again, and they actually recover pretty well. Nucleosomes are not as fragile as we would expect them to be.
Cool. And the time scale for the recovery, for the recoil?
We're talking about like a second, 2 seconds, not a very long time. But we've never done it where like you press and then you do high-speed AFM. I actually do want to do that experiment. It's just technically very challenging because high-speed AFM mode is very different from nano-indentation mode. But that is an experiment that I would like to do to know what is the hysteresis, right? So if you do this kind of deformation, which maybe mimics kinetical forces, that was my idea originally, then does the chromatin look different when you look at the mobility? And that would be a great experiment to do. Yes.
So maybe it's
Angela.
a bit naive, but I was wondering, do you think that the DNA sequence itself could also impact on the elasticity of the nucleosome?
Absolutely, yeah. So that's a great question, and I love that you asked this question. So I think the DNA sequence is fundamentally important for centromeres because DNA sequences at centromeres are AT-rich in almost all organisms that have been looked at. However, neocentromeres, which are new centromeres, they pop up all the time, are not AT-rich. So there's this idea that and this is from Evie Sutoglu's work and others, that the centromeric DNA sequence is Steve published a paper on this as well, is not B-form DNA. It's distorted. And because it's distorted, there's a higher propensity to attract SEMPE. SEMPE seems to like weird sequences. We did work with Case Decker a couple of years ago, looking at the contribution of alpha satellite DNA, which is centromeric, to the stability of SEMPE nucleosomes. It wasn't hugely different, but I think it probably matters from the repetitive nature of the elements, and so that is something that we should be looking at. It's hard to do it in vitro.
I was wondering, when you were comparing the SEMPE from different species, did you take the corresponding DNA, or did you always take...
Right now we're doing alpha satellite, but what we want to do is mouse minor B 120 base pair, because that is the best DNA sequence for centromeres. The yeast DNA sequence is available. It's a unique 120 base pair fragment. The other DNA sequences aren't well known. It's just now the advent of nanopore, so we hope that over time there'd be more sequences available. It's a great question.
Thank you.
Bill.
So about sequences, what do you think about the work from Fukagawa's lab that shows on the Z chromosome in chicken where the sequences are all unique? Centromeres move around.
Yeah. So
they don't particularly need, they're quite happy to shift by 10 or 20 kb, and this
Yeah.
sequence is different there.
I think Glennis's work has turned a lot of this sort of ideas on its head, right? Because what Glennis Loxton's work shows is that centromere is very dynamic. It can move on that alpha satellite array also very dynamically. So I don't actually know what the answer is going to be. I do like that one paper from Oak Ridge where they put in, it was in the chicken DT40 system, I think, where they put in all the different DNA sequences for various centromeres, and then they asked the chicken Sempe, which sequence would it go to? And it was the mouse minor B, which I found weird, like why mouse minor B? So there must be something special. But it maybe it doesn't have anything to do with the DNA, but it has to do with the RNAs that are being made. Perhaps certain RNA properties from these repetitive elements are more likely to fold in a manner that helps SEMPE be retained, or to recruit SEMC, which is what is the case in plants. So I don't have direct evidence that the sequence matters, but I do agree with Angela, it's something we should at least look at to be sure whether it matters or not. But I'm agnostic, actually, at this point. Like, I don't know. Please.
Very interesting talk. So you mentioned supercoiling in conjunction with RNA polymerases injecting.
Yes.
Even before that, when you had these plasmid preparations and you were looking at this mobility, did you see any dependence on what's the supercoiling status of the plasmid and the mobility of these?
That's a really great question. So there's been a big argument in the centromere field whether centromeres are positively supercoiled or not. I think it's just a question of technology, right? So BTMP mapping now exists, which allows you to probe supercoiling. There's Colibacter, this woman, Monica at UW, Monica Guo at UW has developed a method to map supercoiling, and we don't know yet whether supercoiling matters. We did an experiment with Case Decker, where we showed that if you torque the DNA, so it's the same kind of optical tweezer work that Mark showed very beautifully. You can torque the DNA right-handed, right, or you can torque it left-handed, and SEMPE did not care. It assembled just fine on both. But here's the catch, so did H3. So that experiment kind of turned our prediction on its head that I think that nucleosomes can assemble on anything, because eventually when you assemble a nucleosome, this is going to be like boring, but you change the linking number of the DNA, right? So the DNA is constrained in the nucleosome, and then you get supercoils, plectonemes outside that compensate. In vivo, none of this matters because topoisomerase is there. So topoisomerase is going to relieve. Topoisomerase is equimolar with histones in living cells, so it's relieving stress.
The question is also the dynamics of it. Maybe it can, whether you have positively supercoiled or negatively supercoiled can.
We could do it in vitro, but I guess maybe I'm a little reluctant to do it in vitro because I don't know if it matters in vivo. I think that's maybe my only concern. The way I think it might matter is on the loop extrusion models, right? So for big domains where you really can't, topoisomerase cannot relieve like a 1 megabase loop that's right-handed or left-handed. But that's not toroidal supercoiling, that's plectonemic supercoiling, and that's constrained by proteins. Totally different ballgame. So we've not looked, but I think it would be fun to look at some kind of like high-C map to see what centromeres do when you constrain supercoiling.
Thank you.
Great question.
So, it was a very, very nice talk.
Hi, Jose.
I really enjoy it. And first I have a comment, then I have a stupid question. The comment is like, all during, it's just fantastic, and you can see all these things at the same time, mechanical, protein, stuff like that. And it's very hard for me, in a lot of these cases, to figure out what's cause and what's consequence. So do we have, is there more experiment that we can try to figure these things out?
Yeah, so the experiment that we think is a killer experiment is mutating SEMPE to make it H3. Okay. The experiment suggested to me by Eric Green at my site visit, and I was like, it's brilliant, why didn't we think of it? So what you can do is mutate residue by residue SEMPE in the histone fold domain, and we already knew from computational modeling with Garrick which residues matter for that scissoring motion. If we're right, and the computational modeling is right, those 6 residues are all it should take to make SEMPE behave like H3, or H3 behave like SEMPE. It turns out in vivo that experiment's already been done. You can make H3 behave like SEMPE by mutating residues. So we just need to do it in vitro, and that's in the works.
And now it's to the question, are the junk proteins really junk proteins, or that's just our ignorance?
Sorry?
Are the junk proteins that come in, are they really junk, or that's just because we have no idea what they are doing?
Junk proteins, I don't think
you said when you have all these other proteins going.
I think that Bill said that.
Yes.
We haven't mapped, I don't think any protein, you mentioned that you had 4,000 proteins, of which only a few were responsible for chromosome folding during mitosis. But yeah, I doubt that anything is really junk in the genome.
And how about junk DNA? Are they doing anything?
So we don't think there's junk DNA, at least not at the centromere. We know, and this is work from our lab, but also from Beth Sullivan's lab, and Elaine Dunleavy and Patrick Kuhn, and Sylvia Ohart, that the non-coding repetitive RNAs arising from centromeres matter. What was surprising to me is that there's work from yeast, from Karen Yoon's lab, that the RNA matters there from centromeres, which shouldn't, because it's a genetic centromere, it's a point centromere, so I've never understood that data. To me, that suggests that the RNA has a structural role, which is why I asked Bill if there's a structural role for RNAs, but we don't know the answer yet. So no junk RNA, to answer your question. All right?
Thank you very much.
Thank you very much.
So I invite you to continue the discussion with the posters, and I can also smell the dinner. So thank you very much to all the speakers, and enjoy the evening.